The broadcast is now starting. All attendees are in listen-only mode.
Hi, good afternoon, everyone, and Happy New Year. Welcome to the first installment of the HDSA Research Webinar Series for 2017. And today I'm excited to have both Dr. Andy McGarry from Cooper Health in New Jersey, as well as Spiros Papapetropoulos, who is the Vice President of Global Development for Neurodegenerative Diseases and Movement Disorders at Teva Pharmaceuticals on the line. And they are going to be talking briefly about the PRIDE-HD clinical trial results that were just recently announced. But before I turn it over to them, I just wanted to give a quick update to those who may be new to the webinar series about how you can ask questions. So in the menu box on the right-hand side of your screen, you should see something here highlighted in red that says questions. You can type your question in at any time during the webinar, and at the end of Dr. McGarry's presentation, we'll ask those questions on your behalf. We are recording this webinar, so if you have to leave us early or you know others that would benefit from hearing this information, we are archiving this and we'll be placing it on our website as well as on our YouTube channel, which is very easy to find from our hdsa.org website. If you just click here highlighted in red, click the YouTube icon in the middle, and it'll take you to a library of all, not only this, but all of our other research webinars that we've had in the past. Now I'll just mention that the next webinar will be taking place on February 1st. You'll be getting an email notification about that from 12 to 1 p.m. Eastern Time, and I'll be presenting a annual year in review for the past year. And as always, if you have any topics that you'd like to hear more about, send your suggestions to researchupdates@hdsa.org. So with that, I'm going to turn it over to Spiros, who has a few words to say, and then we'll introduce Dr. McGarry.
Thank you, George. Happy New Year, everyone. Dear participants, family members, friends, caregivers, I'm Spiros Papapetropoulos. I'm a neurologist. Trained in genetics and movement disorders. I was previously at the University of Miami and currently affiliated with the Mass General Hospital. For the past 2 years, I had the privilege to work full-time at Teva, leading the efforts in HD and other chronic neurological diseases, working closely with a team of very dedicated physicians and scientists under the direction of Michael Hayden. On behalf of Teva, I would like to extend a warm thank you for being part of this study. You helped increase the knowledge about Huntington's disease and the potential benefits of predopedy. A study would have never been possible without your generous donation of time and effort, and we truly, truly learned a lot. I hope you, your families, and your doctor and your doctors feel our passion for discovering HD treatments or even cures, the passion of the Teva team. You have my personal commitment that the next studies that are currently being planned by Teva in HD will proactively reach out to members of the patient community through both HSG and EHDN to ensure that the study will be even easier to conduct for all and will include fewer site visits, less procedures, and complexity, and more at-home activities. But without further delay, I would like to introduce Dr. Andy McGarry, who will walk us through the results of PRIDE-HD. Assess what dopamine could improve aspects of HD, focusing primarily on motor function, but also exploring a variety of other disease features. Andy is an assistant professor at the Movement Disorders Division at Cooper. He did his residency and experimental therapeutic movement disorder fellowship at the University of Rochester, and he's an active member of HSG, PSG, an MDS member, and a co-investigator for PRIDE-HD North America. Andy?
Thank you, Spiros, and I just wanted to thank you for the terrific message just there to the HD community and for all of the wonderful collaborative efforts thus far through Heart and Open Heart and Pride and Open Pride. It's been wonderful, and we're grateful on the HSG side, and I know that I speak for the HDSA and the HSG and thanking you for all of your efforts, and your passion is very clear, for sure. And I would just parenthetically comment, excuse me, how excited and proud I am to work on Huntington's with colleagues like Spiros and George. You know, there's a lot of people in the fight, for what it's worth, and they're great, talented, and motivated. So it's a complete pleasure. From that perspective, we feel it's very important, again speaking for Spiros and George and the HSG, to have a moment to be able to communicate with you on an easy and useful level, as best as we can help it, regarding things that are typically complex with a lot of moving parts. So we have a brief presentation to help roll through the results of the PRIDE study, to highlight a few things that we want to carry forward that were very interesting. And as Spiros said, we did learn a lot. And the observations that have been made will help craft the forthcoming studies. And so hopefully we can make that clear and useful to you. And as you hear what we're discussing, please feel free, as George had mentioned, to post your comments and questions, and we'll see how we can be of use to you. Next slide, please. And so just to rewind back a bit, the genesis of PRIDE was driven naturally by observations in other studies of pridopidine. And so there were two large ones, as you may recall. There was one conducted by our friends at the European Huntington's Disease Network called MERMAID, and the one that the Huntington Study Group sponsored with Teva called HEART, where over 30 weeks and 12 weeks respectively, there was an improvement in something called the total motor score, which is an outcome measure meant to actually reflect a person's movement. There are ways of measuring that. This is one of them, and it appeared to have some benefit. It's worth noting that in both MERMAID and HEART, that was not the primary outcome measure. And just even to be clear, when you're doing science and testing compounds in people, you have to put your nickel down from a scientific standpoint and say what your primary outcome measure, or the main thing you are speculating is going to change, will be. And defined as it was as the modified motor score, which is a partial version of the total motor score, the primary outcome measure in both of those studies was not met. So from that perspective, they were formally failed studies in that regard. Although when you do that, because it's such a valuable opportunity, you have other outcome measures, some of which are either secondary or exploratory. So you gather a lot of information apart from just that primary outcome measure. And so that's where this total motor score that we have on our slide here is important, because it's one of the things that was noticed to improve. When you see things like that in doing science, it helps you ask questions that turn into subsequent studies, or helps you cast the direction of your thinking for the next round of considerations. It generates new questions. So then PRIDE, in that vein, looked to test higher doses of pridopidine above the 45 milligrams twice daily in a similarly structured endeavor where it's randomized, double-blind, meaning the investigators and you don't know what your assignment is, whether that's the dose or a placebo, and involve a lot of people. So there were 400 participants over 52 sites. We selected people who had a moderate total motor score, that number that would be assigned to their mobility, their movement, slowness, stiffness, things of that nature, was 25, which could be thought of as mild to moderate difficulty with movement. And the main endpoints then for this study, so going back to what I was just saying about a primary outcome measure or a main endpoint, was the total motor score, driven by the things that we're seeing, as mentioned above, in MERMAID and HEART. In addition, something called the Modified Physical Performance Test was added, and that was sort of a nuts and bolts way of looking at how you got on through your day. If you think about the things that happen during your day, walking up and down stairs, manipulating a jacket, picking something up and placing it in a designated place of your choosing, things of that nature are reflected in outcome measures like the physical performance test. It could be thought of as almost a real-world simulation of how someone is able to perform. And so in PRIDE, with those things in mind, subjects were offered the opportunity to either complete 26 weeks, which is how it was originally intended, or complete 52 weeks, which was a decision made in an effort to gain additional information about how people were doing over time, and of course, how the drug performed from a safety standpoint. Safety is always a paramount concern in studies like this. And so when we sat and analyzed the data for all of those things that we checked in the study, all the different testing procedures, the pre-specified outcomes were analyzed as originally intended, meaning the stuff at 26 weeks, because it was originally meant to be 26 weeks. And analyses at 52 weeks for that extended period are formally exploratory. And so they're very valuable, the data that you generate at 52 weeks, but they are just from a scientific standpoint thought of a bit differently than the ones that are at 26 weeks. Next slide, please. And so reviewing the results from a motor perspective, since the total motor score was the main outcome measure, we had good participation. About 80% of people completed the 26 weeks. We had a good cohort, about 2/3 enter the second period, and then we had 58% of people get all the way to the end of the 52-week period. The total motor score showed improvement at all of the doses that were tested. Compared to Heart and Mermaid, it was of similar magnitude. Formally, there was no beneficial effect, which is thought to be related to a large placebo effect that was found. And so what placebo effect is a phenomena where there is a benefit, ostensibly, assigned to an active compound based on a hypothesis you have scientifically that is also seen with a placebo or an inert substance. And so a placebo is not an active substance. It's meant to help you scientifically see differences between active substances and non-active substances. And occasionally, in any domain of science, in testing compounds, particularly in people, the placebo effect will appear. And so that influences how you interpret what you have scientifically. And because there was a placebo effect that was fairly large, it does influence how we think about that seeming improvement in the total motor score for the other doses. So formally, the total motor score did not change across any of the doses that were tested. Now, when you have a large data set like we have in PRIDE, what you will do is look retrospectively, so after the fact, at ways of seeing things in the data that might help you guide additional questions and additional studies. And so if we look at Huntington's disease stage 1, which would be very early, at 26 weeks, 90 milligrams appeared to have a beneficial effect on the motor score. Now that is what we call post hoc, as you see on the slide there. And what that means is you are considering this after you have sort of put your nickel down scientifically about what you think will happen. And so there's an understanding about the extent with which you can be certain about anything analyzed post hoc. But it is, again, valuable because it will help you wonder about important questions downstream or additional things you want to study next. If we continue that line of thinking further and look at HD stage 1, you can see that total maximal chorea and dystonia scores actually did seem to improve at different doses. I have it there for particular doses, slightly granular, but the main point is that some of these other items that were not primary outpoints in the study did appear to improve. So these are important things to consider going forward. Next slide, please. If we shift gears to function, apart from rote motor score, there are other things that are of interest in considering where to go next. One of the outcome measures that we have is something called the total functional capacity, the TFC. And that is an outcome measure that has several domains meant to reflect how one would move through the day and get on with activities that are part and parcel for most people's days. We have a slide coming up where you'll be able to see what it actually is. It'll be a little bit easier to sort of sink your teeth into about quite what the TFC is. But for the moment, the TFC at 26 weeks did not improve formally for any dose. However, if you look at Huntington's disease stage 1 and 2, which again would be earlier Huntington's, there appeared to be less decline at several doses, 45 milligrams, 90 milligrams, and then the high dose of 112.5 milligrams twice daily. At 52 weeks, there was less decline seen in the TFC for the 45 milligram twice daily dose, which is of interest because over a more prolonged period of time, there is post hoc or exploratory analysis data that suggests the TFC may be potentially improved on pridopidine versus placebo. Again, that's exploratory, but that is something that was seen. At 52 weeks, if we look at stage 1 and 2, there was less decline for 90 milligrams twice daily also, and additional coherence between other outcome measures, including the independent scale, which is another way to look at how someone is getting on for meaningful activities during the day, and some other outcome measures that look at how people walk and think for several of the doses. We're going to talk about that a little bit going forward. Next slide, please. And so thinking about the total functional capacity, as just mentioned, this item that appeared to improve at 45 milligrams and 90 milligrams at the full range of the study period, you can see that there are 5 domains. You've got occupation, finances, domestic chores, activities of daily living, that's what ADL means, and care level. And there's a score assigned to each of these domains depending on how someone is doing. This has been established for some time as a very useful tool for assessing how someone gets on through their day. And you can imagine how important things like keeping the house up, maintaining your occupation, vocational difficulties are reflected in that. Finances, of course, are important for keeping the house up, and activities of daily living, certainly. These are things that are self-explanatory by way of bathing, dressing, toilet, and such. And so the TFC is a very valuable tool in that regard. Next slide, please. In continuing to shift gears again towards some of these items that were mentioned outside of the formal motor score and the TFC as just mentioned, ambulation and apathy appeared to improve. There were trends towards improvement in the gait score of the total motor score, so that's a sub-score that involves what it looks like when you walk. Something called the Walk 12, which is a tool lifted from the multiple sclerosis literature, asking several questions about times of the day when you thought you needed to reach out for something for balance, times of the day when you thought your walking was better than not, times of the day you might have fallen, and such. Something called the timed up and go test, which is very simple. You stand up, you walk a certain distance, you turn around, you come back. And then the modified physical performance test, which was the item mentioned at the beginning, that sort of nuts and bolts way of looking at someone's ability to do things. There's a stair climbing task in that also. And so in early Huntington, stages 1 and 2, there appeared to be trends towards improvement. There are also trends towards improvement in apathy for the 3 lower doses, and it was thought to be formally better at the high dose. Apathy is a word for just feeling sort of blah or not feeling interested or engaged in your usual activity. That said, no other specific cognitive benefits or behavioral benefits were demonstrated at any dose through the study. Next slide, please. Importantly, shifting gears to safety, there were no unexpected study drug-related serious adverse events. So just to unpack that a bit, when you do a clinical trial, you're obviously looking very closely at if people have symptoms or feel unwell. And unfortunately, that does sometimes happen. When it happens, the people running the study think about whether that would be related to the compound in a scientific way, whether there's reason to think that, or whether it would be unrelated, as unrelated things can happen while people are participating in studies. And so no events thought related to the drug happened. A dose-dependent increase in something called the QTC was noted. What that is a feature of the electrocardiogram. And so if you've ever been in your doctor's office where they put the leads on you, on your chest, so there's these little tags of adhesive tape on your chest, and there's wires hanging off of them. There's a machine that generates usually a pink and white piece of paper. It's got squiggly lines on it, and it's meant to reflect how your heart is working. That's an EKG or an ECG, an electrocardiogram. And if you look at that, there are features to that squiggly line, and those features are expected to fall within certain shapes and durations. So what that squiggly line reflects is the movement of electricity through your heart, and that movement is meant to happen at a certain rate. And there are acceptable rates for the different bumps on that squiggly line. And so the QTC is an interval between two of those bumps, and as the dose increased in the pride opening study here, that distance also increased. Now, why that matters is because sometimes when the QTC is too short or too long, the heart can beat funny, and you can have problems with maintaining the flow of blood. But what's very important is that even though the QTC increased in a dose-dependent way, there were no associated adverse events. So in other words, the people who had this happen were not reporting, say, funny heartbeats or passing out or anything of that nature. So there was no correlation between adverse events and this observation on the EKG, the lengthening of the QTC. There was no effect on blood pressure. There was a dose-dependent increase in heart rate that also did not have any adverse events tied to it in a notable way. No notable lab abnormalities were noted. There were behavioral events, psychiatric events reported in all arms. These are things that are expected to be part and parcel with Huntington's disease. They didn't appear to be particularly more overt or egregious in any of the study groups. Irritability was the most frequently observed event, and it was more prevalent in the placebo group, as it turns out. Depression and suicidality were reported in all the arms. They weren't reported in again, any frequency or severity thought to be meaningfully impactful for conduct of the study in the end. The Columbia Suicide Score, which is a means with which to measure this, did change from negative to positive, meaning that someone behaviorally was not feeling their best. That was reported in all arms. And the rates of these things are consistent with the known natural history of the disease. So in short, from a safety perspective, things that were expected did appear. They were not present in undue or egregious amounts or severity. Next slide, please. And so just to summarize this formally, the PRIDE study was a negative study for this pre-specified primary endpoints of the total motor score and the physical performance test. There is some suggestion of benefit on motor performance in early disease, as we mentioned. So when we think back to the stage 1 and stage 2 groups that were analyzed, it appeared that there was benefit that might be an item of hand going forward worth a closer or additional study. In particular, there was some suggestion of benefit on the total functional capacity, that outcome measure that we showed you that has the different domains in it reflecting someone's ability to meaningfully get through their day. At 26 weeks, there was a trend seen, and 52 weeks, there appeared to be formal exploratory benefit. Again, exploratory meaning you're looking at it after the fact, so it's thought of differently than primary outcome measures. Because the primary and secondary outcome measures were not met, but there is suggestion of benefit in other exploratory endpoints, there appears to be good reason to have further study. And that's something that going forward is being planned, as Spiros had mentioned. We did learn a ton in the conduct of this study, and lots of interesting things to consider going forward. With that, I'll stop for a moment and ask George and Spiros if they have additional comments or observations they'd like to make. I appreciate everyone listening. I know that that's a lot to hear at once, so thank you for your attention and looking forward to your questions.
Thanks, Danny. Great job. I have nothing to add on my end. Anything from you, Spiros?
Nothing, George. I would like to extend a warm thank you to Andy and the wonderful overview of the data we received. I greatly appreciate it, Andy, and looking forward to working very closely with you in the near future.
Likewise. Thank you, Spiros.
I mean, so just to, and folks, if you do have any questions, please do feel free to type them in now. There is one that's come in, and it's kind of the obvious one that everyone I think is thinking is, basically what's next? Is Teva considering a second clinical trial? Will it, and will that start, or I think there's probably referring to the stages, stage 1 or 2 of HD will involve just stage 1 or 2 HD patients based on your results from Pride HD.
I can certainly take this question, George. Thank you. It's a very reasonable question, and the answer is unequivocal yes. We're actually very actively in planning stages for what we call a phase 3. So this is the last phase of clinical testing before a drug gets approved. So based on the results of the next study, we will be looking, if results are supportive, to be filing for marketing authorization. The study will be global, and based on the results that we've observed so far, yes, the focus will be in stages that are supported by the Pride HD data set. So we're actively looking and discussing with experts in the field and regulatory authorities. Globally, we're preparing our documents to submit so the study can start in a timely fashion. And based on these discussions, we will finalize the stages of the disease that we're going to be focusing, but we will definitely focus on the science and follow the data, in this case, that are supportive of early, but earlier stages of Huntington's disease as a potential target for predominant maximum benefit. The time frame, because people may be wondering about the time frame, again, there are many unknown factors as you're initiating a large phase 3 program, but the goal of the Teva team and the group of scientists and clinicians that has been assembled to operationalize and bring the study to life is to initiate it as quickly as possible.
Thanks, Spiros. That was another obvious question. You've handled that second question for everybody in terms of timing. One question about the placebo effect. So you guys, can you speculate on why there was such a large placebo effect shown in the Pride study, and what can be done in the upcoming phase 3 study to prevent this from happening again?
That's an excellent question, and I'd be curious about Spiros' thoughts as well. The placebo effect is just something that is built into the fabric of studying human beings. It just is always going to be there and always something you have to think about. There are ways to minimize that potentially depending on how it is you're formally observing somebody. And that can be a bit less relevant depending on what your outcome measure is. So when you think about placebo effect in the total motor score, you're relying on someone who is expert and trained to look at a person with Huntington's disease and match their performance to a predefined set of ranges for severity. Right? So there's numbers that correlate from absent to severe, and you have to find that number based on what you're looking at, and then sum it, and then there's your score. So there's some interpretive license in the course of doing that. That's just part of how we do things. And we minimize that as best as we can with a uniformed outcome measure and a uniform set of standards for the people who conduct studies. So these are highly qualified people who have to pass a series of tests to demonstrate that they're somewhat coherent with how experts would otherwise perform. So we try to minimize it as best as we can. Whether there are other ways to do that will be of interest in the future. I don't want to speak for Spiros or Dave at length. He'll certainly have some thoughts about this, but I know that other places are and other means of investigation are being developed to try and actually quantify how it is that we measure people. So that might end up influencing the data that we get and how we think about the performance of compounds and clinical trials in the future. The world at large is becoming more digital. It has for some time, so it is an expectation that how we study compounds and clinical trials will also follow suit. Going forward, it is a thought always about whether placebo would interfere with what we saw. So I think we'll try to be creative going forward about how we try to minimize that beyond what I said. First, turn it over to you. I'd be very curious what you think too.
That is an excellent segue. I just want to highlight a couple of things. We are going to be practical. We did identify some study design elements that could have contributed to the placebo response that we observed, and we're going to modify them in the next study. We are also putting together a panel of world experts to help us further investigate the root causes of the placebo effect in this particular trial, and we're going to be using the collective knowledge and applying it directly to the phase 3 study. So yes, our goal is to control the placebo effect to the extent possible. Controlling the placebo effect and the expectation bias that is introduced by the anticipation of benefit is something we are also going to be focusing on, and educating our investigators and our study participants and their families on the principles of clinical research. And this has demonstrated in the past that it could contribute to modifying this placebo response. So it is a series of learning exercises that we're going to engage the scientific community, some technical modifications that we're going to be making to the actual study design, and a series of educational attempts on the principle of clinical science for patients, their families, and investigators. So this is our plan.
Thanks, Spiros. A couple questions are coming in about how people can participate in the phase 3 study when it is announced. One question is, will the phase 3 study be open to people that did not participate in the previous studies, Pride and the Mermaid and Heart studies?
I would recommend talking to your treating physician to further evaluate the eligibility of individual patients and participants for this particular trial. We are still in the process of developing the detailed list of subject and participant inclusion and exclusion criteria or eligibility criteria. We're going to be discussing these criteria both with the patient community, leadership of scientific bodies in Huntington's disease, as well as regulatory authorities. So the criteria at the moment are not fixed, and we will certainly circle back, and perhaps, George, we can have another webinar when the time is approaching for study initiation to provide more insights around this criteria.
Of course, yes, and it's kind of a nice segue to say that, to let everyone know that we here at HCSA and other organizations are in close contact and communication with the folks at Teva, Spiros, and Andy, and others, and when those details are provided, we'll make sure that we share them with the community, both through things like this, through webinars or convention, but also the local sites, when they are announced for this upcoming phase 3 study, will be all listed on our new clinical trial matching service called hdtrialfinder.org. So, when the information is finalized, we'll make sure it's out there in an easily digestible forum for families to get and try to get in contact with their local sites to see if they're eligible. So one question, it's kind of, this is probably for Spiros, and folks are wanting to know, what else is Teva working on for HD?
So Teva has a commitment to HD. This stems out of through Michael Hayden's leadership. We're actively working on the SD809 FDA application. So this is a program that came through the acquisition of Auspex Pharmaceuticals, and SD809 is a deuterated form of tetrabenazine that successfully completed clinical development. We remain very optimistic, and we are striving to get SD809 to patients as quickly as possible. This is one major program. Other than predopidine, we are also evaluating laquinimod, which is an anti-inflammatory agent that is also being developed for different forms of multiple sclerosis in the HD population. The trial is actively enrolling participants. It's a phase 2 study, therefore a learning study based on which we will have an opportunity to assess next steps for laquinimod in HD. We expect that we're going to continue enrolling for laquinimod HD, new participants globally for at least the first half of 2017.
Thanks, Spiros. One more question has come in. When will subjects who participate in the PRIDE study be told of their treatment assignment?
That is something that we need to further look into, perhaps I'm taking down good notes here, so I have my to-do list following this meeting, and we will get back to our patients and their families with an answer to this question, perhaps in our next webinar.
Okay, thank you, Spiros. There are no other questions, but I just want to take this time to thank both of you for joining us and presenting this important information on this important trial to the families and HDSA. And we look forward to working with you and helping you in the execution of your phase 3 study for predopidine. So with that, I will end this call, and please make sure that you join us on February 1st for the next research webinar. And we look forward to talking with you soon. Take care.
Thank you, George.
Bye-bye.
And Spiros, thank you very much.